best.
Great. Thanks, everyone. Thanks, Margaret. Paul Arthur here, the lead for low methane livestock. And as most of you have worked out, most of work area deals with genetic improvement. Started from the bottom up, back again. Started from the bottom up as in what we hope to accomplish in this area. Basically, the beef and also the sheep industry continually providing commercial producers with breeding stock, which are genetically low methane producers, but more importantly as well, highly productive. So with that, I'll go on next, please, to the beef project, which I lead. And simply put is to work on, and it also implies to most of the other projects, our learnings from NLP which firstly indicates that methane emissions are heritable, and secondly that if we have the opportunity to have the rumen microbiome data, it improves the accuracy of the prediction of the genetic merit of the individual animals that we produce. So based on that, next slide, please. We are taking opportunity in this project. We have a current Southern multi-breed bean project happening where all the production traits are being measured on 5 breeds of cattle: Charolais, Angus, Shorthorns, Herefords, and Wagyu. We have Brahmans in there because there's a complementary project in the north, so we've got a genetic link in there. And the plan is to measure methane on those, and not just methane, but also CO2 and oxygen. CO2 is very important, especially where in pastures where we cannot measure feed intake per se. Research done indicate that it's a good proxy at a genetic level for feed intake. Next, please. And I think that's it for one of the beef components in the area. The other one is over to you, Sam.
Thanks, Paul. So this is really building on what Paul discussed. He's building essentially a reference population for predicting seed stock as well as commercial animals for reduced methane emissions. So a portion of this project, which I'm leading, is a collaboration with Angus Australia to aim to record more Angus animals so that they can build a genomic reference population for those individuals in the breed. If you go to the next slide, please, Margaret. So the main genomic reference population would build on past work that was done as previous projects, as Paul noted, combined in with the multi-breed herds that Paul told us about for Southern multi-breed, and record an additional 2,000 records for Angus animals. There's a direct impact in industry from this project because the Angus Society has genotyped large numbers of animals through their breeders, and this information that we record in the project for reduced methane can be directly applied across to those breeders, which will help their breeders select young seed stock for reduced methane, as well as select young commercial females for reduced methane. And given especially that some recent analysis has shown that the cow herd has 48% influence of Angus genetics, this is a large number of commercial animals that could potentially be accessed and influenced by selection going forward. That's all. Thanks, Margaret.
Julius.
So this is the same proposal, but then for sheep. So we want to see how we can breed for more methane efficient sheep. So next slide, please. Sorry, here I work with Hutton Oddy, who did a lot of prior work on it, and Daniel Brown and other people also in Egbo and sheep genetics to implement breeding values. And we've had experience with that system before. So next slide, please. So it's we really want to look at methane efficiency in the sense that we want to reduce the amount of emission output per product produced. And of course, there's a system efficiency, but we're going to focus on individual animal variation. That doesn't automatically mean that you want to look at methane per head, because with breeding and selection, it might be more efficient to increase or decrease the mature weight of the ewes, or to improve the reproduction efficiency of ewes. That might have a bigger effect than just a CH4 per head. So we want to put things in context and do some modeling first, and even before submitting the proposal, some work has been done, but to get it totally clear what is the best approach. Then second is, if we do measure methane output, what is the best trait to measure? You can measure it in lambs. That would fit our current setup with the MLA resource flock, but it might be much better to do it in ewes. And we probably do a bit of both just to see what the genetic correlation is. It's also important to realize that all those traits are correlated to each other. So if you breed for lower methane emitting sheep, what is their growth efficiency and their reproductive efficiency and their wool production efficiency? So all the things hang together, and we have to get good parameters to make good decisions in the breeding program. But previous work has been done, and we assume that we're going to measure methane emission in a whole bunch of animals so that we can use genomic selection, because it's hard to measure those traits on farm, and it's hard for breeders to breed for it. So as Sam said, we have a reference population. We need to measure a few thousand animals for the phenotype as well as genotype, so we can predict new animals' breeding values. We've had that model before with eating quality and the sheep CRC. We have selected from traits that are not measurable on farm, but we have a reference population and turned that information into breeding values for breeders, and they're now heavily using that in the lamb industry. And we consider, but we're not fully sure yet to what extent, to do additional support work in the microbiome, mostly to see if that helps to predict better breeding values. Next slide, please. So the plan is really first look at modeling and literature to see what the best approach will be. Then the idea is that we are measuring many animals per year, lambs and or ewes, and at least 1,000 per year. So in a few years, we have a decent sized reference population. We're going to genotype those animals and then develop genomic prediction and see what accuracy that gives. And that could produce ASBVs, and then we're going to optimize the breeding program to see what is the best approach to select for more productive use, yet to lower methane efficiency per product output. So the budget is a bit unclear, but most of the money will be spent on measuring lots of phenotypes and some on genotyping. And the microbiome work, we have to define it a bit better, and talk to some colleagues, but that will be supportive work. Thank you.
Well, thank you. So my name is Gene Tyson, and I've recently moved to QT, where I've established the Centre for Microbiome Research. And really today what I intend to tell you about is a project that we're proposing as a platform technology that, based on some of the presentations I've seen today, could be useful across a wide range of different projects, everything from additives and supplements through to feedstocks, and of course within this program in livestock. And we've already seen within this program that there's a number of microbiome studies that could really be facilitated through the technology that I'll present to you today. And so what my group does is we're focused on using high throughput sequence-based technologies to explore microbial communities with the ultimate goal of doing targeted cultivation of these organisms in order to develop novel probiotics or live biotherapeutics. Next slide. So the way that my team works and what we have done in the past across a range of different systems, including many systems involving methane production and consumption, including thawing permafrost in northern Sweden, through to the discovery of organisms involved in anaerobic methane oxidation, to some work that we're doing within the human microbiome now, which is very analogous to the program that I'll talk about here, which has been successfully commercialized, is that we use these high throughput sequencing technologies to get a fundamental understanding of these microbial communities at both the species and strain level. And we do that through a combination of omic technologies, including metagenomics, where we reconstruct genomes of the organisms within a microbiome sample. We overlay that with functional activity through metatranscriptomics and proteomics, which tell us about which genes are expressed under different conditions, and then tie that into metabolites that are being produced by those microorganisms, which ultimately impact the phenotype of the animal. Now, in the process that you'll see today, we're going from this bulk community scale right down to single cells. And we go down to these single cells because this allows us to isolate these organisms and use them as probiotics within the context of comparing high and low methane producing animals. And so the flip side of this, of course, is that in order to do this analysis and to do this analysis well, we've had to develop a range of different bioinformatic tools to analyze these communities with intricate detail going right down to the strain level, which has not been possible in the past. And so the tools you can see here help us analyze this data, make sense of it, and work out what genes are enriched and depleted under different conditions. Now, the reason this becomes useful, next slide, is because when you understand the community differences between these organisms, you're able to use a big data-driven approach to go and isolate specific organisms that are providing a functionality that's important to either transitioning a pathway away from methane production or helping the performance of the animal in terms of weight gain, etc. What you'll see here is that we've developed a range of different techniques that are allowing us to isolate previously uncultivated organisms. There's a few IP sensitive technologies that I won't talk about today, but the one I'll present here is one that we've been developing for the human microbiome, which is a process called anaerobic MADFAX. And this stands for multi-angle detection fluorescence activated cell sorting, with a specialized sorter that we're getting custom built in the US to allow the cultivation of organisms in a strictly anaerobic environment. What we're able to do is we're able to see individual cells within a community using the fluorescence activated cell sorting. With a combination of metagenomics, we can then work out where every single species is within a community and target them for specific cultivation. So when you tie together the information coming from a large data set of animals, both high and low methane production, you're then able to specifically target organisms that you believe are going to have a benefit to the animal, both in terms of reducing methane and or animal performance. And so then from this, you're able to sort those individual cells and do high throughput isolation of these organisms for then testing in a large number of animals. Now, as I said, I think this ties into a wide range of different projects, and I'd be very happy to talk about collaborations with anyone who's on the call today about how we might use this technology. Thank you.
Okay, Ben, do you want to have any input, Ben?
Yes, I do. Good
Ben has
morning or good afternoon, all. So I'm just going to give you a brief rundown of our project in this area. And this is a collaboration between DAF Queensland, UQ, and a commercial partner. And yesterday with Paul and Sam, we discussed extending that to take advantage of the project Sam's looking at as well. So I'll talk about it in terms of the products that are going to come out of the project. The first product would be a saliva test for methane emissions. And this is based on profiling the microbiome through the saliva, because through various work that's been done, there's a pretty strong correlation between the microbiome in the rumen and the saliva, because cattle chew their cud. So that's an easy sample to collect and a fairly easy strategy to profile the microbiome. And we've also done enough work now ourselves that's been recently published, and enough work's been done overseas as well, that by integrating that information, we can recognise low and high methane emitting microbiome profiles. So in summary, the product is a saliva test that predicts methane emissions with some accuracy. So it's like prediction of a phenotype. And that could be used in the breeding herd and also in feedlots as well. And we talked yesterday about taking advantage of the work that Sam's doing to extend that to southern cattle, really, because our work is focused mainly on the north. The other component is we've talked a lot through this meeting about anti-methanogenic compounds and delivering those in extensive environments. The other component of this is working out when in the year is the best time to deliver those, given that in Northern Australia, there's enormous variation in both abundance of the pasture and the nutritional content of that pasture. So there's a big team behind this, and some of them are online. If they very briefly want to add to what I've said, feel free to jump in now. No, sounds like they're happy with what I've said, so I'll throw it back to you, Paul.
Okay.
Thank you.
No, thanks everyone. As you could see, the summary slide that you can see there, we've got the key thing is the resource we use, and the animals are very well characterized. And from the projects mentioned, we have 2 sources of well characterized cattle, and also existing one in sheep, which we could use. Secondly, the fact that those traits of importance are going to be measured in those projects, and then we superimpose on it methane and methane related type traits. Yes, we open for collaboration in relation to microbiome. We've already got 2 groups within the low methane group with the capability, and we talk to anybody else as well. Now, what we need to do is to look at more synergies, not only within this area, but other areas. The other thing that I have just needs to mention, most of you are aware, is that for genetics improvement, we already have an existing conduit for breed plan for cattle and sheep genetics for sheep. And so we'll be working with AGBU and the necessary authorities to go through that. But in between that, there is a lot of R&D underpinning what we have mentioned in terms of refining technologies, etc., to achieve it. And that's it for